Welcome students! As I have promised you earlier to do EmSAT sample questions from the MOE, this is the continuation from sample 1, which you can check out the link in the description down below. And that's what we will be doing today. So make sure you like and subscribe down below and turn on the notification bell to receive many more videos like this one from Mrs. Biology. Let's begin. Let's start. John's disease is a chronic enteritis that is caused by pathogenic bacteria. The bacteria grow inside the ileum of ruminants. Which statement explains what happens? Let's read. Maybe you will tell me, I don't know what is John's disease. I will tell you we will follow the logic in that. Let's read the first option. The production of protein slows down as the bacteria grow and divide. Hold on. We know that the bacteria participate in the production of certain enzymes, proteins. So, logically speaking, once the number increased, the number of bacteria increased, sure the production of proteins will increase. So, A is wrong. Moving to B. The intestinal lining thickens, resulting in decreased absorption of nutrients. That makes sense. Why? Because most, if not all, most of the infection by bacteria, especially in the gastric, will cause diarrhea. What does that mean? That means the intestine will not absorb the nutrients very well. But let's read the rest of options to clear our doubts. C. The bacteria stop the production of amylase inside the small intestine. Hold on, this is wrong. Because amylase enzymes produce inside the mouth. Sure, now A and C are wrong answers. Move to D. The movement of chyme in the small intestine slows down as the bacteria grow and divide. Again, the same idea. Bacterial infection mostly will cause diarrhea. So once the diarrhea is happening, that means the chyme will be very fast when they are in the small intestine because there is no absorption. And that will support that idea of answer B. That means the answer here is what? B. Let's read the second question. To investigate the effect of light on the growth of plant shoots, 3 shoots were used under different conditions as shown in the apparatus setup below. Which is correct about the growth of the 3 plant shoots after 1 day? Let's read. The first one. The high concentration of oxygen on the shaded side of the untreated shoot has stimulated the plasma membrane's proton pumps and reduced the pH level, causing cells to elongate. As a result, the shoot grew toward light. That's right, 100%. This is the right procedure. Let me show you the animation. As you see here, that's exactly what will happen. Because amount of oxygen on the shaded area will be high, so elongation of the cell because of the pH level will decrease. What will happen? The plant will grow toward the light source. But let's read the rest of the answers. B. The foil, the second one, has inhibited the ion uptake into the cells on both sides of the shoot due to low pH level in their walls, causing cells to stop dividing. As a result, the shoot with foil cap did not grow. And that's wrong. Why? Because the impact of the foil cap is to prevent the sunlight. And we know without sunlight, no photosynthesis. And without photosynthesis, no nutrient. That means there is no growth. So B is wrong here. C. The increased concentration of oxygen on the shaded side of the untreated shoot has elevated the pH level in the cell walls. I will not continue because it will not elevate. It will decrease the pH because of proton pumps will be stimulated. So C, sure, it's wrong. B and C till now are wrong. D. Exposure to light destroyed the oxygen in the tip removed shoot, causing the cells at the tip to stop growing. As a result, the shoot with tip removed did not grow. Hold on. Once they removed the shoot, that means they removed most of apical meristem tissue. What is apical meristem? It's like a stem cells that will differentiate and enhance the growth. So once they remove it, the growth now decreased or even inhibited. So D also is wrong. So the right answer here is what? Is A. Let's read this question. Which statement is correct about the thickness of uterine endometrium during a typical menstrual cycle? Before that, come with me and let's look at these two graphs that related to hormone levels directly with uterine cycle, the thickness I mean of the endometrium. Okay, as we see here, estrogen and progesterone levels. Estrogen will increase and the thickening of the endometrium will increase. During the ovulation, estrogen level will decrease, but who will increase again? Progesterone, and that will cause the endometrium to be more thick, thicker. Okay, let's go back to the options. An increase of progesterone concentration results in thickening of endometrium after ovulation, which is right 100%, as I showed you before. Please go back and check the graphs. B, around day 21 of menstrual cycle, the endometrium gets thinner due to an increased production of estrogen. This is wrong 100% because it will continue and become thicker. So B is wrong. C, the concentration of estrogen falls immediately after ovulation. That's right. Causing the endometrium to become thicker? Sure not, because who will cause it to become thicker is the progesterone. Okay. D, if a pregnancy occurs, the levels of estrogen decrease and progesterone increase, causing the endometrium to become thicker. Hold on, this is talking about pregnancy, and the question is asking about what? The menstrual cycle. So the right answer here is what? Is A. Moving to this question. The diagram below shows the oxygen hemoglobin dissociation curve. Which of the following changes in CO2 concentration and pH level would probably be responsible for the shift from X to Y? As we see, look at the graph, we can see the dissociation process is happening. Okay, now we want to choose from A to D, the right one. Let me talk with you about how the carbon dioxide impact or affect the pH level of the blood. We all know that the carbon dioxide influences the pH of blood by reacting with the water inside the plasma, and that will give us the carbonic acid. Now, this carbonic acid will dissociate to form hydrogen ion and the hydrogen carbonate. So increasing the hydrogen ions inside the blood will decrease the pH. So here we will look at what? When the carbon dioxide level increase, what will happen with the pH? Will decrease. Let's move. A, increase, the CO2 carbon dioxide will decrease the pH level, and this is right 100%. But let's see other options. What will happen with the dissociation? Decrease, sure not. B and C talking about the decreasing of carbon dioxide, that means this is wrong. Here, it's wrong. Moving to D, increase in carbon dioxide, will increase the pH? Sure not, because that will increase the hydrogen ion, and the hydrogen ion will lower the pH, will not increase it. It will be acidic medium. So the right answer here is A. This question looks easy. Which of the following is a potential safety hazard in the laboratory? Let's read. A, handling hot test tubes or glassware with tongs. That's right. That will help in achieving the safety. So A is wrong. B, shutting off the gas and electricity in case of fire in the laboratory. Again, this is one of the steps that we have to follow to achieve the safety. So B also is wrong. C, pouring an acid into water inside a fume hood. Also, that's right, because we need a fume hood in order to absorb all the fumes, the toxic or the bad fumes, in order not to be affected. So A, B, and C, we have to follow them to achieve the safety. D, using a strong base to neutralize an acid if it splashes onto the lab bench, and this is disaster. Maybe will lead to burn or something else. So the right answer, we mean its hazard, is D. Now this question. Which of the following is an example of a dependent variable? That means in any experiment. Let me just remind you, by independent and dependent variables. The independent variable is what you change, and the dependent variable is what changes because of the independent. Let's see here. A. The amount of fertilizers added to measure the growth of different plants. This is the independent. Fertilizers here is the independent, not the dependent that they are looking for. So A is wrong. B. Measuring different trees to see which one is the tallest. Actually, this is not dependent or independent, because here I'm looking for the results. So B is wrong. C. The amount of sugar that dissolves completely at different temperatures. I think this is the right answer, because amount of sugar here is the dependent. Depends on what? The different temperature, and the temperature here is the independent. So C is the right answer, but let's look at D. The amount of air pumped into a balloon to make it fly. So where is the dependent or the independent variables that support which answer? C. So the right answer here is C. This is a nice question. The diagrams below show the results of an experiment in which two microbial species were grown a specific culture medium. Which of the following interactions suggests the interdependence relationship between the two species? Let's analyze the graphs. The first one, the species A grown alone in a culture medium, and as you will see, because of the nutrients available, so the increasing growth is happening. Let's look at B, also by themselves in a culture medium, also they are increasing. But when we put them in the same medium together in one culture medium, we notice that A keep growing and increased, but B with time died, decreased. So for answer A, is it a mutualism? Sure is not. We have to focus here on the third graph. Why? Because mutualism, both of them will grow and no one will be affected. So A is wrong. B, competition. I think this is the right answer, because they will in one culture medium, so they will compete the resources, the nutrients, but A, it seems stronger. But let's continue. Commensalism, also not. What is commensalism? One will get the benefit and increase, and the other one will not be affected, but that did not happen. So also C is wrong. Moving to D, predation. No way. What is a predation? Predation, if A eats B, but actually A is not eating B, A is growing in a culture medium. So the source of a nutrient is not the B. So D also is wrong. For that, the right answer is the competition, which is B. An evolutionary question. Let's read it. The table below shows the frequencies of red and white flowers in a small population. Over a period of time, a random change has occurred and the frequencies of this population has changed. Which of the following evolutionary processes explains what happened to this population? I have to search for the key term before reading the options. And the key term here is the random change. I'll tell you why. Let's start with a natural selection, the first option. What is a natural selection? Is the process through which populations of living organisms adapt and change. So it's not randomly happening. B, artificial selection. What is artificial selection? Is when humans breed plants and animals for particular genetic characteristics. Also, it's not a random change. What about genetic drift? Is a random process that can lead to large changes in populations over a short period of time. It seems that this is the right answer, but let's go to gene flow. What is a gene flow? I know it's like a migration that describes mechanisms of gene transfer from one population to another, as I mentioned. So what applies to this question, I think, the genetic drift, because it's a random process. So the right answer here should be C. Let's read this question. A dichotomous key was created to identify a specific organism. According to the characteristics below, Which of the following animals belongs to annelids? First of all, a dichotomous key. It is a scientific tool used to identify different organisms based on organisms' observable traits. Okay. They are talking about what? Annelids. Annelids, we all know they are talking about the worm. Listen, if you don't know the organism that they use this tool with it, I'm sorry to tell you, you will not answer it in the right way. But at least learn with me how we can answer this question or how we can use the dichotomous key. A. These annelids shows bilateral symmetry, so we can go to B, or shows radial symmetry. Sure, they show the bilateral. Bilateral, that means if you have an imaginary line, you can have same two halves of that organism. So they are a bilateral symmetry, so we will go to B. If it wasn't, it was radial, we will go to C directly. So because we will go now, because it's bilateral, we will go to B. B. They reproduces asexually, that means it's animal 1, or they reproduces sexually, we have to go to C. Listen, for annelids, they have both reproduction organs. Male and female. They are considered as a hermaphrodite. So they can reproduce sexually. Maybe they can reproduce asexually, but once they have the male and female, so we will go with the sexual reproduction. We will go to option C. C. They have this annelid has a hydrovascular system? Sure not. They have a closed blood circulation. So I have to go to option D. Again, I told you, you have to look at the observable trait. If they have it, characteristic, you will proceed to that option. Now, I am in D. They are hermaphrodite. Yes, 100% they are hermaphrodite. They are not a fully functioned male. I told you from the beginning, they have male and female organs. So they will consider them as a hermaphrodite. So the right answer should be what? Animal 3. What does that mean? C is the right answer. Here a blood inheritance question. An Rh positive man whose parents were both homozygous for Rh positive marries Rh negative woman. Seems here we have typo, it's woman, not woman, whose father was heterozygous for Rh positive. That means he gave her the Rh negative, not the positive one, once we have two alleles. Now, what percentage of their children will be expected to have homozygous Rh positive blood? Let's focus on the term homozygous. Her father, or their kids, the ones we are talking about the children, their father positive and their mother negative. And each of the parent will give one allele. So sure, the dad will give the positive once he is homozygous Rh positive, and his wife, their mother, will give them sure negative. So how it will be homozygous? It should be 0. But to clarify it more, come with me. Let's continue here. The father is homozygous for Rh positive, and the mother, she is negative for the Rh. So their kids, when they will donate for them these alleles, will have Rh positive, but not homozygous, heterozygous. Actually, I did a video about the blood transfusion. Please refer to that video. It will be in details in the explanation. So let's go back and choose the right answer. So what is the percentage of their children will be expected to have homozygous Rh positive blood? Sure, it will be 0. They will never have homozygous, they will have heterozygous. So the right answer is A. This is a good question. Let's read. Which of the following structures are present in photosynthetic prokaryotes? Photosynthetic prokaryote, the prokaryotes that can use photosynthesis to make their foods. And directly, without thinking, we should think about cyanobacteria. Okay, looking at the table, we will see that they are looking for mitochondria, chloroplast, DNA, and cell wall. Without thinking, we all know that prokaryote, like bacteria, they don't have any bounded organelles. And mitochondria also chloroplasts are considered as a bounded organelle. So if you depend on just this information, you will find that A is the right answer. But maybe you will start asking, Miss, you are talking about photosynthetic, so photosynthesis, and we need the chloroplast because they contain the chlorophyll. Hold on. Chloroplast, yes, in eukaryotic cells they contain the chlorophyll, but in bacteria, you will find the chlorophyll in the cytoplasm. Okay, so no need for a chloroplast. And even the mitochondria, no need, because the energy inside these prokaryotes, I mean the bacteria, because of the different gradients of ions. So the right answer without thinking, it should be A. Yes, they contain the DNA, they have the cell wall as a structure, but sure they are missing what? The mitochondria and the chloroplast. Regarding B, C, and D, all are wrong answers. So the right answer here is A. This is a pedigree question. Let's read. The pedigree below represents the inheritance of a human disease over 3 generations. What is the probability that the daughter of individual 2, generation 3, would receive the disease trait from her father? Let's look. So this is generation 3 and this is individual 2, this man. And he is unaffected. And unaffected male, sure. In order to decide the right answer, first of all, I have to know, is it dominant or recessive? And one of the main rules of pedigrees, if we did not skip any generation and all affected, I mean at least one in each generation, that means it's a dominant disease. And as it's clear, yes, it's a dominant disease. What do you mean by dominant here? If we have 2 alleles, if both of them capital, I mean dominant, or at least one of them, I will receive the affected person. But what we see here, he is already unaffected. So he should be recessive. Let me clarify it more now. Come with me. Here, as we agreed before, that this disease is dominant and each trait needs 2 alleles. So let me assume we have the letters B. So B capital is affected person, or B capital B small, because one allele of dominant, it's enough. That's why I told you the genotype for the father is recessive, B small, and we all know one of the parent will give only one allele. So the chance to give his daughter a dominant allele is what? 0. So the right answer is what? Let's go there. It should be 0. So the right answer here is C, because in all means he will give her a recessive allele. This is a genetic question. Listed below are the percentage composition of each nitrogenous base from DNA of 4 species. What is a valid deduction from these data? It's very clear. We have the percentages for adenine, guanine, thymine, and cytosine. For sea urchins, yeast, maize, and the human. I cannot decide exactly what is the relation here till I read the options to see which is the right statement. A. The structure of DNA in maize is similar to the one in the animal cells. Animal cells, I have here a human and I have here sea urchin. But if I pick any percentage, for example, guanine, 22.8, but in a human it's a 20.7 as a percentage. In sea urchin it's 17.2. Any other one? Here, thymine, 27.2. In sea urchin, 32.8. In a human, 30. So this is a wrong statement. B. The amount of purines is always greater than the amount of pyrimidines. First of all, purines representing adenine and guanine, and pyrimidines, thymine and cytosine. Just let's look at the amounts here. They said purine greater than pyrimidine. Purine, I mean the adenine, here 26.8 for maize, for example, but the thymine 27.2. So directly, this statement is wrong. C. Adenine shows a complementary base pairing with guanine. Come on. Yes, they are purine,
But adenine all the time pair up with thymine by 2 hydrogen bonds, and all the time guanine pair up with cytosine by 3 hydrogen bonds. So also C is wrong. It seems the right answer is D. The 4 bases of DNA in all species show complementary base pairing, and that's right. All the time adenine with thymine, as I mentioned, and guanine with cytosine. And we call this rule Chargaff's rule. We call it Chargaff's rule. So the right answer here is D. This is a question. Which of the following statement is correct about the effective role of enzymes in facilitating chemical reactions? Let's see the options. A. They provide energy to the reaction to speed up the conversion of reactants to products? Sure not. Enzymes, they facilitate, they will not add or remove anything from the chemical reaction. And we all know what is the main role of enzyme. Let's see the other options. B. They speed up the chemical reaction rate by lowering the activation energy. This is the right answer, but I will continue with you the rest of options. We all know activation energy is the minimum energy needed to start the chemical reaction. So once the enzyme is there, this the enzyme will minimize this activation energy, so the chemical reaction will speed up. C. They raise the temperature of the reaction to allow more reactants to convert into products? Sure, no. D. They activate the chemical reactions by increasing the level of activation energy, which is wrong against what is happening. Let me just display this graph. This is the energy and the progress of the reaction. The dot lines representing the enzyme work. After adding the enzyme, see the activation energy with enzyme, it's very low if you compared it with the activation energy without enzyme. So, sure, the chemical reaction will be faster. For that, the right answer is B. What about this question? Which of the following orders represents the secretory pathway of proteins into the extracellular environment? It seems an easy question. It's talking about the proteins, and we all know proteins synthesized inside the ribosomes. And because we know the ribosomes located in the cytoplasm scattered there or on the rough endoplasmic reticulum, directly I will cancel options C and D, because you should know that the smooth ER function is to make lipids, cholesterol, steroids hormone. So I'm happy that I have only 2 options right now. Rough endoplasmic reticulum, Golgi apparatus to process and modify the proteins, to Golgi cisternae, then secretory in vessels, then to cell membrane to be exported outside the cell. This is a good option. B. Rough endoplasmic reticulum, Golgi apparatus, Golgi cisternae, then lysosome. Hold on. Why lysosome? Lysosome contains digestive enzymes. So this is very wrong, not only wrong. Let me just give you this picture and look at the pathway. So from rough endoplasmic reticulum, the protein will enter the Golgi body or Golgi apparatus to be processed. Then it will be in the vessels that will be exported outside the cell. So let's go and choose our right option, which is the first one, rough endoplasmic reticulum and Golgi apparatus, cisternae of the Golgi, secretory vessel, then the cell membrane. Let's read this question. A student was observing a specimen at a total magnification of 100x. What does that mean? That means he is using the objective lens of 10x. Why I'm telling you this? Because we have 2 lenses, the ocular lens, the eyepiece, and the objective lens that is very close to the slide. Now, 10 times 10, the objective lens magnification times the ocular lens will give me the total magnification. Let's continue. While observing, he moved the objective lens to what? To 40x. So if in your mind right now you are calculating the total magnification, it will be 40 times 10, the ocular lens. So it will give me 400x. Now,
After doing that, he focused on the specific region of the specimen using the coarse adjustment knob. He did not use the fine adjustment to fix the resolution. What problem would the student face? Let's read. A. The slide would probably break. Makes sense, and I will show you that in the figures. B. The specimen will not be in the field of view. I doubt. He is already, as you see here, focused on the specific region, so I think B is wrong. C. Resolution power will increase tremendously. How? And he did not adjust it. He was only using the coarse adjustment. Let's see D. Light intensity will be higher than expected. Why? He did not mention in the question that he is using the light knob, I mean the condenser. Let's see these pictures. As you see here the 10x objective lens, and here the 40x. And we can notice that the 40x objective lens is taller. Okay? Is taller. Moving to this figure, can you see? So it will break the slide. So if we want to choose the best answer, I am supporting the first answer, that the slide would probably break. This is the last question. Which of the following occurs when electrons flow through the electron transport chain during cyclic photophosphorylation? From its name, that means it happens during the photosynthesis process in plants. Let's read the options. First of all, synthesis of ATP, which makes sense, release of oxygen, and we know it's one of the products of photosynthesis, and reduction of an ADP to an ADPH as an energy carrier molecule. Let's see this. We all know that the type of photophosphorylation usually occurs in the thylakoid membrane. The electron begins in a pigment complex called photosystem 1 in a cyclic electron flow, then further passes through primary acceptor and pyridoxine and the cytochrome. As we see here, we have the ATP synthase, this enzyme that will use the energy from the flow of hydrogen ions to add phosphate group to ADP, adenosine diphosphate, to make ATP. I think the another figure and animation will make it more clear. Again, this is the electron transport chain in the thylakoid membrane. As you see here, the stroma, and this is the photons, the energy that gained from the sunlight. Here, the photosystem 2, photosystem 1, the cytochrome, and the major thing that will make the ATP synthase enzyme. So, if we want to answer this question, sure we will go with the first option. What about releasing oxygen? Releasing oxygen, it happens because of oxidation that happens to water molecule. And regarding the reduction of an ADP, it's not a phosphorylation actually. It's a reduction, as you see, adding hydrogen ion. So the right answer is synthesis of ATP. Now, we have completed these questions, and this may be the end for now in EmSAT questions. However, don't worry, as I am still awaiting new sample questions, so I can continue this series to have more sample questions coming up for you students to watch. So, stay tuned to receive the next part of these EmSAT sample questions by liking, subscribing, and turning on the notification bell down below to receive the next upcoming videos from Mrs.Biology. Thanks for watching.